So we are talking about human developments and genetics. So till here we completed third stage, second stage, and first stage. So how the delivery will happen, labor will happen. We studied in detail. So this is very, very important. And all all medical or non-medical have to be this knowledge, because in everyone's life this incident will happen. So the basic knowledge is no, okay, to protect our ladies, to protect our females, okay, uh from becoming severe. Okay, so it is our duty. So let's start. So here we have third stage we completed. Now we have the infant at birth. So after delivery, what will happen? Okay, this is very subjective, because everyone's after everyone's in everyone's life this incident will happen. The baby will come to a new world. Okay, so at that time all the family members, including her mother, father, everyone is happy. But what medically the baby is happening internally, so let's talk about that. Immediately after delivery, the umbilical cord is clamped and cut. So you know that clamping will happen. Okay, one scissor is that will cut, or in with the uh, needle, with the plastic clamp will given, which is very clean, sterile. Okay, and we'll cut it. And the infant's nose and mouth are aspirated to remove any fluid that might interfere with breathing. So whatever fluid in mouth, in nose, it will be cleaned with the suction or aspirated with the help of small nozzle, anything, whatever the doctor decide. Now the infant is independent of the mother, and the most rapid changes occur in the respiratory and circulatory system. Now the after delivery, the after birth, baby doesn't need mother circulation and mother respiration. He can breathe with his own respiratory system. He can circulate his blood with own respiratory system. At the level of CO2 in the baby's blood increases, and respiratory center in the medulla is stimulated and brings about inhalation to expand and inflate the lungs. Full expansion of the lungs may take up to 7 days following birth, and the infant's respiratory rate may be very rapid at this time, as high as 40 respirations per minute. So basically, in first 7 days, it will take lungs to expand, because now the lungs are very in calm position. It is not functional. Yeah, okay, their structural are developed till 9 months. So what will happen? So now it will take 7 days. But for that reason, the respiration is very rapid. Abdominal also, we can see that abdominal in and out. Okay, so and all the respiratory muscles, including chest, whatever is that, will expand. More expansion is there. Okay, breathing promotes great pulmonary circulation, and the increased amount of blood returning to the left atrium closes the flap of foramen valve. The ductus arteriosus has begun to constrict, apparently in response to the higher blood oxygen level. Full closure of the duct atrios may take to 3 months. And so here we have the heart. Heart is very important, because after birth, some some babies have, we listen that they have hole. It will take 6 months, 12 months to close the hole. So this is the atrium. At the atrium, we have the foramen ovale. So you can see that foramen ovale. So here we have patent foramen ovale. You can see that this is the septum. This is the septum. This is the hole. Okay, blood will come left atrium, and right atrium. It may be close after delivery. It may be during 3 months. Okay, it will take 3 to 6. Some babies will take 3 to 6 months. So after this closure, they have separate uh, left ventricle, left atrium, left ventricle, right atrium, right atrium. If the closure doesn't happen, so the from the left atria, the blood will mix here. Right atrium, the will blood mix here there. So deoxygenated, oxygenated blood mix there. So we call septal defect here. Okay. Next, we can see that this is the fetal circulation. You can see this. Okay, fetal circulation in this image. This is the ductus arteriosus. You can see this is ductus arteriosus. Okay, and here we have ductus. You can see ductus arteriosus. Okay, this is the block will close. Okay, this is also be closed, and venous also closed. Everything is closed because here in the during 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 pregnancy, the all the circulation is coming from mother, so there is no need of functioning heart. Okay, heartbeat will there, but their function is to separate the blood from arteries, veins. It's not from left atrium, left ventricle, right atrium, right ventricle. Okay, so it doesn't separate. So basically, the oxygenated blood will come and it will deoxygenated will blood grow through the fetal circulation and from the umbilical cord to the mother. Then mother will do the deoxygenated blood from to the oxygenated blood convert and it will again deoxygenated blood. So there's a reason heart septa is not fused properly because it is now non-functional. Now, after birth, the foramen ovale, ductus arteriosus, ductus venous, all will be bruised and uh, it will have to be doing pain because you know the baby is the ductus venous no longer receive blood from the umbilical vein, okay, and begins to constrict within a few minutes after birth. It will have to be constrict within few minutes after birth, or within a few weeks, the ductus venous become non-functional ligament. So there is no work of ductus venous. Now the ductus arteriosus also constrict, okay, and it will constrict and it will close slowly it will close up to 3 months. The infant's liver is not fully mature at birth and may be unable to excrete bilirubin efficiently. This may result in jaundice, which may occur in as many as half of all newborns. Such jaundice is not considered serious unless there is another possible cause, such as RH incompatibility. So here, most of the infant, almost all the infant doesn't have after birth, the liver is not completely functional. So that's the reason not functional, not matured. Okay. So what will happen? The bilirubin, what is you have to excrete from the body will not happen. So there is a reason the amount of bilirubin is high. So baby will looks in pale in color. So there's a reason they will give light therapy. Okay. And advice to keep the baby in sun. Okay, so these are the reasons. So this is all about human development. So now we will start genetics. Okay, genetics is the study of inheritance. Most, if not virtually all human characteristics are regulated at least partially by genes. We will first look at what genes are, then describe some patterns of inheritance. So genes is nothing by genetics. Genetics is the study of inheritance. So whatever you are getting from the family, So it is the study of genetics, genes. So these are responsible, whatever similarity you are getting, whatever things you are getting. So it is related to your family tree. Okay. Now we will discuss about genes and everything. Each of the cells, each of the cell of an individual except mature red blood cells and egg and sperm cell contains 46 chromosomes, the diploid number. These chromosomes are in 23 pairs called homologous pairs. So basically in the each cell we have chromosomes already discussed in the part cell. DNA have chromosomes. In the chromosomes we have 46 chromosomes. If we paired it, we will call diploid number. So, and we will call 23 pairs, we named is homologous pairs. What we named? Homologous pairs. One member of each pair has come from the egg and is called maternal. And the other member has come from the sperm is called paternal. So one member from the mother, one member from the father. If it is coming from the egg, we will call maternal. If it is coming from the father or sperm, we will paternal. Maternal and paternal. The autosomes are the chromosomes pair design 1 to 22. The sex chromosomes from the remaining pair, in women these are designated XX and in men XY. So basically we symboled sex chromosomes of men with XY. We design female sex chromosomes with XX. Okay, understood. Chromosomes are made up of DNA. Okay, you know all bit DNA. This have DNA, protein. The DNA is the hereditary material. So in the chromosomes we have DNA and protein. So it will give the information about your hereditary, your family, your tree. You may wish to refer chapter 2. So already discussed DNA. That's one again we can see that DNA here the image is given or not. Probably not. So let's see one more time about cell and internal structures. Hmm. This is image is very good. You can see that. So you can see this image. This is the cell. In the cell, you have the nucleus. In the nucleus, you have chromosomes. In the chromosomes, we have DNA. In the DNA, you have nucleosomes. Nucleosomes, this is the gene. You can see this gene. Gene have this type of things. Guanine, cytosine, adenine, thymine. So this is well structure with the gene structure with the this is DNA. You can see that in the chromosomes we have DNA. So let's come to the topic. So DNA is the main thing which have hereditary information. Okay, the sequence base of in the DNA of chromosomes is the genetic code for proteins, structural proteins as well as enzymes. So already we discussed this is the names of the all the genes we have, that this are the proteins, structural proteins, guanine, cytosine, adenine, thymine. Okay. For example, a specific region of the DNA of chromosome 11 is the code for the beta chain of hemoglobin. Because an individual has two of chromosomes 11, he or she will have two genes for this protein. A maternal gene inherited from the mother and a paternal gene inherited from the father. This is true for virtually all of 20,000 to 25,000 genes estimated to be found in our chromosomes. In our genetic makeup, each of us have two genes for protein. So basically here in the chromosomes we have 53 paired chromosomes. If we go 11th number chromosome of RBC or hemoglobin, okay, what will happen? In that 11th chromosomes, we have two genes, one from maternal, one from paternal. So likewise, if these genes, we will take mother sample, father sample, we will see that these genes and that genes are same or not. Then we will get that this is the hereditary tree or family tree located belongs to this boy belongs to this. These are the fathers and mothers. Okay. Genotype and phenotype. What are genotype and phenotypes? Let's come discuss. For each gene of a pair, there may be two or more possibilities for its expression. Okay, how it will appear or how it will affect the individual. These possibilities are called alleles. So basically, every gene has expression. So like we are expressing, actually we are not expressing, our genes are expressing that. Our genes are expressing that. So those expressions are two types. It may be how it will appear first thing and how it will affect individual. These are the two things will in the scene in expression of the gene. Okay, so those things are combinedly called alleles. What do we call alleles? A person therefore may be said to have two alleles for each protein or trait. The alleles may be same or may be different. So alleles will call it two alleles because one gene have two alleles. So each gene have two genes have four alleles. So likewise, each gene have two alleles. Okay. If the two alleles are same, the person is called homozygous. If the two alleles are different, we will call heterozygous. So if two alleles, two expression, the genes expression are same, what we will call homozygous. What we will call homozygous. If two expressions, two alleles are different, we will call heterozygous. Very important. Okay. For genetic, the genotype is the actual genetic makeup, that is alleles present. The phenotype is the appearance or how the alleles are expressed. When gene has two or more alleles, one allele may be dominate over the other, which is called recessive. For a person who is heterozygous for a trait, the dominant allele is the one that will appear in the phenotype. The recessive allele is hidden but is not lost and may be passed to children. For recessive trait to be expressed in the phenotype, the person must be homozygous, recessive, that is, have two recessive all alpha the trait. I think it is not required, but I will explain you because here that if the allele also belongs, two things here, which expression is dominating. If is homozygous, we will call which is dominating, we will call recessive. Dominating. If the one allele is dominating another, we will call recessive. Okay. So what we'll call? We will call homozygous recessive. And if it is not dominating, they are working parallelly. So here we will, what we will call? We will phenotype or genotype. Okay, they are makeup like that. Their alleles are present and they are expressing in same way. Okay. So if we add homozygous recessive or heterozygous recessive, here one example is given. An example will help to put all this together and illustrate. When doing genetic problems, a Punnett square is used for show the possible combination of genes in the egg and sperm for a particular set of parents and their children. Remember that an egg or sperm has only 23 chromosomes and therefore has only one gene for each trait. In this example, the inheritance of eye color has been simplified. Although eye color is determined by the many pairs of genes with many possible phenotypes, one pair is considered the principal pair with brown eyes dominating over blue eyes. A dominant gene is usually presented by capital letter, and the corresponding recessive gene is represented by same letter but lowercase. Okay, so here everything is giving. I think it will cause some confusion. So I think we have to uh, stop here only. So, but I will explain this thing because uh, the gene which is expressing now, so it will give that expression. Okay, so here example is given that eyes. Some eyes are in brown color, some are in black in color, some are blue in color. Okay, the iris. Okay, the iris, the color. Here, some are dominating. They are look like blue, but dominating brown color. But they are black in color, but dominating blue color. So how this is happening? Because those genes are expressing their work to express the color. Okay, their genes are expressing their color, but some genes have dominating effect, recessive effect. They are not homozygous, they are heterozygous, they are different. So only black color you will get homozygous. Okay, but they are heterozygous there. Each no, not they are homozygous, they are heterozygous, they are expressing, and in the expression we have the different genes are expressing their own, but some are dominating. Some are dominating. So that's the reason they are looking blue along with black in color. They are looking brown, but they are black, but they are looking brownish color because they are dominating. Okay, so this is because of the genotype, phenotype, and heterozygous and homozygous. Okay, inheritance. So likewise, if the gene or if the newborn have one gene, but one gene is coming from the father or parent, but the born gene is not dominating. The gene which is coming from the father, coming from the paternal maternal, is dominating. So likewise, the baby is coming with the symbolic presentation of his father or his mother. Understood? So that's the reason why walk like your father. They walk like their mother. They talk their voice like that. So because of the gene expression, dominating gene expression. Okay. So here the example is given: gene from brown eye color, gene from blue eye color. Okay. So BB same will cause brown in color. If BB is coming brown and blue both, if B is more dominating here, either both are same. Okay, either B this B is dominating. So here blue color, blue brownish color. If this BB is dominating, so what we will call blue color eyes. Okay, this is very important. So in some cases we have multiple alleles. Multiple alleles. So this is very important. Multiple expressions, multiple expressions, two, not two, more than two, more than two. The best example of the pattern is inheritance is the human blood type A, B, O group. For each gene of this blood type, there are three possibility alleles: A, B, or O. A person will have only two of these alleles, which may be the same or different. O is the recessive allele. A and B are co-dominating alleles. So I think you understood what is the uh, main thing what we have to know. These are the examples. There can be multiple alleles. Here we have the inheritance, sex-linked traits. So I think so. They are the related to the genetic things. So sex traits like the problems from the from the father, from the mother. So it will also transfer to the baby in coming age if he is doing the same things what it did father do, like drinking, smoking, the gene will be activated and the gene will do harm to the baby and they have same hereditary diseases. Okay. So they are linking together. I think so. I think the base of genetic we understood. Mostly the whatever genes are they are dominating. So there's the reason our symbolic representation. The mother symbolic representation will come to the baby and he will be present like that. His face, his color, his walking style, his talking style. So likewise, the diseases also transfer from the father to the baby. So this is the thing you have to understand. Base core is this is the very important. I think you understood. So we completed the part of genetic and human development. Next, we have the microorganisms, microbiology. So I think it is, we will discuss in microbiology subject. It is not related to anatomy and physiology, but is related to the microbiology. So we completed anatomy and physiology after taking long time. So I thank everyone. So okay, so we completed essentials of anatomy and physiology.